Hello, everyone, and welcome to Chapter 3 of Human Physiology. To kind of catch you up on where we are right now, Chapter 1 focused on a lot of introductory physiology stuff. We learned all about the levels of biological organization, among many other things in that chapter. And essentially what we're going to do for the rest of this first unit is make sure that we kind of have a fundamental grasp of some of the lower tiers of those levels of organization. In the previous chapter, Chapter 2, you learned all about atoms and molecules. Well, in this chapter, we are going to focus on the next tier up. We're going to focus on cells and what cells are constructed out of and specific cellular processes. So this first video, we are going to focus on the cell membrane, and then we will start to move into heavy concepts regarding membrane transport, and then you're going to see on the next slide that most of some of the basic organelle biology is going to be on your own work. So if we look at some of your on your own responsibilities for this chapter, I'm going to cover the vast majority of all of the membrane transport processes in this specific lecture video series. I'm not specifically going to talk about bulk transport, talking about exocytosis and endocytosis. That's easy enough to handle on your own, and you probably talked about that plenty in your general biology class that you've already taken by now. Cell organelles, just make sure you get a good appreciation for the general functions of the nucleus, both the rough and the smooth endoplasmic reticulum, the Golgi apparatus, and lysosomes and peroxisomes. So as long as you just have a general understanding of what those organelles are for, that should be good enough. But for the nucleus and the ER in particular, there is going to be a little bit of extra information that you'll need to know regarding how protein synthesis works. The cytoskeleton, microtubules, and actin, just make sure you have the most fundamental base understanding of what these are for. I'd be surprised if you see more than one question about these on the exams. Either 0 or 1 question, so not super duper duper important. They are important to cell biology, but there's not really going to be that many times we talk about them in the context of specific physiological processes. And then your major responsibility for your on your own work is making sure that you've got the protein synthesis process down. So this begins with transcription, the synthesis of messenger RNA molecules using a DNA template. Those mRNA molecules have to be exported out of the nucleus through nuclear pore complexes, and then the translation process begins, in which ribosomes capture those mRNA molecules, and working together with tRNAs that carry amino acids and rRNAs, which make up the most important parts of the ribosomes, they work together to synthesize proteins amino acid by amino acid. So just make sure you go through that and make sure you have a pretty good understanding of that. I will talk a little bit more about that later on in this chapter when we talk about genetic diseases and mutations, but make sure you have a look at that before you get too far in this video series. And then the very last thing here is chromosomes and the cell cycle. Now, for the cell cycle, I'm not expecting you to be able to recite for me word for word all the different things that happen during the cell cycle. So interphase, prophase, metaphase, anaphase, telophase, cytokinesis, and all that good stuff. Just make sure you've got a good general understanding of what the cell cycle is for, what mitosis accomplishes, the major differences between mitosis and meiosis, why mitosis is good for what it does, and why meiosis is good for sexual reproduction. I'm not going to ask specific questions about what happens at anaphase 2 or what happens at this specific event during cytokinesis. So I'm not going to ask those sorts of questions, but just make sure you've got a good understanding of what those things are there for and what they accomplish. Okay, so the first thing that we want to discuss is what makes the cell functionally able to participate in many physiological processes. And the answer to that is complicated, but the first place for us to start is going to be the cell membrane, or the plasma membrane as it is also called. So the cell membrane is a physical barrier that is going to separate bodily fluids into 2 different compartments, right? You've got your extracellular fluid, which is all of the fluid outside of cells, and then you've got your intracellular fluid, which is all of the fluid inside of your cells. So we've got two different fluid compartments that are separated by cell membranes that every cell in your body has. If you look at this illustration, you can actually kind of see what we're talking about here. So you see various different cells which make up some tissue. We're not necessarily specifying in this picture, but you see in yellow here, we've got the intracellular fluid, the fluid inside of cells. In purple, we've got one of our types of extracellular fluid, which is called the interstitial fluid. That is the fluid between cells, but fluid that is not in blood vessels. And then in the pink color, we've got the blood plasma, which is a type of extracellular fluid that is contained within blood vessels. Now, those are not the only two types of extracellular fluid. We could also talk about lymph and cerebrospinal fluid and things like that, but these are most definitely the two major types of extracellular fluid that we will need to be aware of moving forward. Okay, so the cell membrane separates bodily fluids into two different compartments, but the thing to understand about the cell membrane is it's not like it's a concrete wall. It's not some impenetrable barrier. So the more correct way to describe it would be to say that it is selectively permeable. It lets certain things pass from compartment to compartment while restricting the passage of other things. It picks and chooses what moves from one fluid compartment to another. So in this way, for that to be possible, the cell membrane needs to be both dynamic and pliable. It has to be able to adapt to changing conditions, and it has to be fairly flexible. So the ability of molecules and atoms to pass from one fluid compartment to another, this is going to be a fundamental need for being able to maintain homeostasis in the body. So if the cell membrane were some brick wall, if it were some impenetrable barrier, that would be very bad, and being alive is not something that would really be possible at that point. So there are all sorts of interesting conversations that we're going to have in the future regarding the importance of membrane transport processes and why those are important. But before we get too far ahead of ourselves and before we put the cart ahead of the horse, we need to examine the specific structure of the cell membrane so that we can understand why the cell membrane is not impenetrable and why it is instead selectively permeable. And in doing so, we are going to have to borrow some terminology that we looked at in the previous chapter. All right, so the picture that you're looking at is a specific type of lipid molecule. This is a phospholipid. So on your own work in the previous chapter, you looked at various kinds of lipids. You looked at triglycerides, you looked at steroids, you looked at prostaglandins, you looked at waxes and things like that. A phospholipid most closely resembles a triglyceride. To be more precise, a phospholipid is actually a diglyceride. You'll recall that we have a glycerol molecule, which is basically going to be our carbon skeleton here. This is not a true polymer. We can't attach as many things as we want to it by dehydration synthesis. We can only attach up to 3 things. So a triglyceride would have 3 fatty acids attached to the glycerol backbone. A diglyceride, hence the name, will only have 2. So a phospholipid, being a diglyceride, only has 2 fatty acid tails. That third position, which could be taking up another fatty acid, is instead taking up a molecule called phosphatidylcholine, which is just represented here by the negatively charged phosphate. So this actually creates an interesting situation. So the phospholipid is interesting because it has very different chemical properties depending on which part of the molecule that you're looking at. We have a word for this. This is called an amphipathic molecule. One part of the molecule is very, very hydrophilic, the part up here, which is represented by the big red circle, what's called the hydrophilic head. That phospholipid coupled together with the glycerol is very, very polar and very electrically charged. So it's hydrophilic because water can form hydrogen bonds with it, if you will recall. Whereas the bottom part of the molecule, made up by the very hydrophobic and non-polar fatty acid tails, that forms up the hydrophobic part of the molecule. So in the previous chapter, we got some practice identifying molecules. This one is hydrophilic, this one is hydrophilic, this one is hydrophobic, this one is hydrophilic. And we did that by looking at the molecule and just using some common sense and looking at functional groups, this one can interact with water, this one cannot. Well, a phospholipid has two major parts to it. One can interact with water and the other cannot. So that lends to a really interesting question. How is a phospholipid going to behave if we introduce it into an aqueous environment? Now, the thing to consider is that we are constructing an entire cell membrane. So we're going to use lots and lots of these phospholipids like little building blocks to make our actual cell membrane. We're constructing a cell membrane out of these phospholipids. So in trying to determine the best way to build a cell membrane, we need to consider how these phospholipids are going to interact with water. The best way to make our membrane is going to be finding a way to make everyone happy, to get the hydrophilic heads interacting with water, which is what they want to do, and prevent the hydrophobic tails from having to interact with water, which is something they do not want to do. Okay, so let's try one way of making a plasma membrane. Let's just take a single layer of phospholipids and just stick them together like what you see here. Okay, so what's the problem here? You've got these heads pointed out towards the extracellular fluid, and you've got the tails pointed down towards the intracellular fluid. What's the problem with that? Well, if you have been paying attention up until this point, you know that these red heads here are hydrophilic. That is where you find the polar heads. So they're going to be perfectly happy. They're up here interacting with all of the water that is in the extracellular fluid outside the cell. The tails, on the other hand, are very hydrophobic, and we are forcing them to interact with all of the water that you find in the intracellular fluid. Don't forget, even though the solute composition of these two fluids are very different, the water composition is exactly the same. So each fluid that you see here has an exactly equal percentage of water there. So the heads in this case are very happy, and the tails are very unhappy. So we need to do something about that. This is clearly not the best way to go about making a cell membrane. The correct way to make a cell membrane is by making a bilayer of phospholipids, where you have a top layer and then a bottom layer that is flipped upside down right underneath it. So if we instead use a bilayer, everyone should be happy. The hydrophilic heads will point outwards towards the fluids, outwards towards the extracellular fluid and all the water there, downward towards the intracellular fluid and all the water there. Meanwhile, those hydrophobic tails point inwards towards each other, and you do not find any water at the middle of what we'll call this phospholipid sandwich. Water out here, water down here, but no water here in what we call the hydrophobic core. This is another example of a hydrophobic interaction. It's a very, very, very stabilizing interaction that arises from hydrophobic molecules interacting together so they don't have to interact with water. Very stabilizing, and a good reason why the cell membrane holds together as well as it does. All right, so another thing, now that we've covered that, another thing for us to consider is the necessity for the plasma membrane to be very fluid. Remember, we said it needs to be dynamic and pliable. So this is going to be a crucial aspect of allowing certain molecules to pass through. Again, if this is just a brick wall, nothing's going to get through. But if it's fluid, things can kind of squeeze their way through. So since we are dealing with lipids, specifically phospholipids, let's put this in terms that everyone who's ever spent any time in a kitchen can understand. You've got two major types of fats that you work with in the kitchen. You've got the solid fats like butter or lard, and then you've got your oils like olive oil or vegetable oil, canola oil, peanut oil, whatever. So you know the difference, right? Butter and lard, those are solids at room temperature, right? So you're dealing with an actual solid gelatinous substance. Oils are fluids. They're kind of viscous fluids, but they are still liquids at room temperature. Both of them are lipids. They're both pretty similar in terms of their molecular and chemical composition, but one's a solid and one is a liquid. Now, we do not want our membrane, which don't forget, our membrane is made up of lipids here. We do not want our membrane to be solid. We don't want it to be like butter or lard, because if that were the case, it really would be an impenetrable barrier. We want our membrane to be much more closer to like an oil, like a fluid. And the membrane does take on what we might call a semi-fluid appearance. And this happens for two major reasons. We avoid being like butter or lard, and we are closer to being an oil for two major reasons. Number one of those two fatty acids that are attached to the glycerol and the phospholipid is of the unsaturated variety, and this produces a kink in the fatty acid tail that causes any adjacent phospholipids that are right next to each other to have to kind of space each other out. Let me go back and show you exactly what we mean here. If we go back and look at the phospholipid structure, you've got two fatty acid tails. One of them is saturated, meaning that it contains the maximum number of hydrogen atoms possible, and it basically forms a straight line. Whereas the other fatty acid is of the unsaturated variety. Having a double bond here reduces the number of hydrogen atoms in the backbone of the molecule, but it also angles the rest of the fatty acid tail away from the other one. So this causes the phospholipid to take up a little more lateral room, and because of that, we can't pack these phospholipids together. A big reason why butter and lard are solid is because they have very high density. We're able to squeeze those lipid molecules and pack them together into a very small space. If you're dealing with unsaturated fatty acids, that's basically like sticking your elbows out. People can't get very close to you if you've got your elbows stuck out to each side. So this is going to reduce the density of anything that contains unsaturated fatty acids and cause it to be much more fluid, meaning that it can't really become solid as easily. So that is reason number one why the cell membrane is a semi-fluid. You've got those unsaturated fatty acid tails. The other reason is that cell membranes contain another type of lipid that you learned all about in Chapter 2, which is cholesterol. Cholesterol molecules actually wedge themselves in between phospholipids, and that also spaces the phospholipids out and keeps the melting temperature of the membrane right where it's supposed to be. Now, cholesterol gets a bad rap, and we'll talk about this again when we talk about steroid hormones in a couple of chapters, but cholesterol gets a bad rap. Everyone just assumes it's bad, but you absolutely have to have cholesterol. It is absolutely essential for many physiological processes. Every cell in your body has to have cholesterol in its cell membrane, or things are going to go really bad. Okay, so that's going to do it for this first video, which introduced the cell membrane. Next time, we will take this discussion further, and we will actually use what we just learned about the cell membrane and its structure to learn about function. How does what we learned lead into how molecules and other sorts of materials can pass through the membrane under different circumstances? All right, well, thank you for your attention, and I will see you in the next video.